Hello Year 12. Today we're going to carry on looking at our work on the nervous system. So you looked at the structure of neurons and the layout of the nervous system last lesson. So today what we're going to do is look at the concept of what's called resting potential. So you need to be able to describe in a sequence how resting potential is maintained and state the role of channel proteins and the sodium potassium pump and the uses of ATP, and then we are going to be looking at graphical source of information and organising your own notes, and obviously creating your own notes. So most of this session is going to be me talking through key ideas. Obviously you can pause this at any point, make notes, rewind, go forward as you like. There is a slide later on with some tasks I would like you to do, but most of this is me going to be talking through the process of generating resting potential from which you can then create your notes. Okay, so key thing about resting potential is it's a voltage. So the potential term here refers to potential difference. So it's the charge across the membrane of the axon, or in fact the whole neuron, because remember if it's a sensory neuron we might be talking about the dendron, but quite often if you look at textbooks it just talks about the axon, but it's across the whole membrane. And in fact even when you start looking at receptors, you'll see there that they may well have a charge. In your heart muscle, their heart muscle cells are also charged and polarized. So even though we use the term axon, it means that any part of the neuron that is conducting or is able to conduct a nerve impulse or action potential. So now where the evidence for this came from was from some scientific investigations into squid. Okay, and they have a very large axons called giant squid axons. It's not necessarily that the squid are enormous, it's that their axons are very large, which means that they are useful to use in research because you can insert electrodes into them relatively easily. These are unmyelinated axons, so they're very large to speed up nerve impulses. Remember we talked about diameter being important in speed of nervous conduction, and if neurons are unmyelinated, the best way to get speed is to make your neurons wider. Okay, so what they found was that when they inserted an electrode into the axon, so across the plasma membrane or cell surface membrane of the axon, and had one electrode outside and one electrode inside, they found that the inside of the axon, the axoplasm, and therefore the membrane, was negatively charged inside compared to outside. So there was a potential difference of voltage across the membrane of about, well, it says I've got here between minus 60 and minus 70 millivolts, and that's because different textbooks and different organisms have different resting potentials. It doesn't vary within one neuron, so 1 minute it's minus 60 and another minute it's minus 70. It's at a value, so you might see it written as minus 65. Okay, but for whatever that organism is or whatever that neuron is you're looking at, it has a particular resting potential. And when it's at this resting potential, we say that it's polarized. Okay, so key term is this idea of polarized or polarization. So if you remember that, that'll be useful. So what we need to understand today is, well, how is this resting potential created? Because we talk about the idea that your nervous system being an electrical system, but actually it's not really like the same as electricity, which is to do with the movement of electrons. It's actually to do with the movement of positive ions that create this. And you're going, positive ions, we get a negative charge here, so how does that work? So we'll talk you through that. Okay, so key things we need to be aware of. It's about an imbalance of concentration of positive ions across the membrane, and this creates the resting potential. The cell surface membrane or the plasma membrane is really important here. Remember, you've got the phospholipid bilayer, and the phospholipid bilayer is impermeable to ions. So if ions are going to move across the membrane, you're going to need to use carrier proteins or channel proteins to do it. So we've got a little bit of a link here to your foundation biology, thinking about the structure and the fluid mosaic model, how that could be brought into questions associated with this. Okay, so you've got lots of sodium potassium pumps embedded into the axon membrane. Okay, so these are active transport protein pumps. that move sodium and potassium ions across the membrane. I'll talk to you about the direction in which they're moving them in a moment. But it's an active process, so it's using ATP from respiration. Okay. There are also lots of channel proteins for two different ions, sodium ions and potassium ions. And most of these are what we call voltage-gated channel proteins. In other words, they are like gates, so they can be open and closed, and they close or open depending on the potential difference across the membrane or the charge. And if you think about protein structure, remember you have got that importance of charge in protein structure and tertiary structure, which can affect the shape. So how these open and close depends on the charge, and that affects the shape of the protein temporarily. So they either open and allow ions through, or they close and don't. Okay, so let's have a look at a little animation on the next few pages that a couple of slides that are going to take you through this. Obviously you can pause here if you want to make a couple of notes. So here we've got a representation of a part of the axon. So this is only showing one side of an axon, if you think about it. And just draw a little rubbish diagram. Okay, so imagine I've got, here's my cell body, here's my axon. What type of neuron am I drawing? Whee, high quality. Okay, there you go. So motor neuron, cell body, dendrites at the other end. Here's my axon. So what we're really looking at here is one side, the cell surface membrane. So we've got the tissue fluid on the outside here, and then the axoplasm. Okay, the cytoplasm of the axon there being shown down here. Okay, so in the membrane you have got a number of different proteins, as I've already mentioned. You have got sodium potassium pumps, potassium voltage gated channels, and sodium voltage gated channels. Okay, so they sit in the cell surface membrane like this, creating channels. Okay, so what you will see then, I mean these are diagrams, not really what they look like. Okay, so at rest, which is a bit of a misleading statement as we'll see later on, at resting potential, which is this voltage, the sodium voltage gated channels are closed. Okay, so here's our sodium voltage gated channels. You can see the gates are shut, so ions can't move through. But most of the potassium gated channels are also closed, but some of them remain open. So the membrane is therefore pretty much impermeable to sodium ions, but more permeable to potassium ions. Now also, which I haven't shown you here, there are chloride ions and charged proteins, but they are unable to move across the membrane. So there's no system for moving them across the membrane. So I've missed them out of this diagram. They appear on the next one. Okay, so let's look at what's going on with the resting potential. So here is the same diagram, but I've now populated it with, this is a sort of non-polarized neuron before anything's happened. Imagine how we create this from the start. So what we've got here, we've got some chloride ions, here's some chloride ions, we've got some negatively charged proteins inside the axon. So they are the negative charge, and then we've got positively charged ions here. And you have to imagine that there's, at rest, there's an equal number of positives and negatives on each, when there's no charge on the axon, it's all equal. It doesn't add up if you look at it, but it doesn't really matter for this. Okay, the key thing is it's not about these negative ions at all, it's about these positive ions. So the sodium potassium pump is going to actively transport sodium ions out of the cell and potassium ions into the cell, and this pumping is uneven. Okay, so you saw there 2 potassium ions were pumped in, but 3 sodium ions were pumped out. So you're already now going to start to have an imbalance of charge, because we've moved 3 positives out but only put 2 positives in. So we're creating a charge. There's more positive on the outside than there is on the inside already, but we're also creating a concentration gradient as well. Okay, so we're getting higher concentration of sodium outside the cell and a higher concentration of potassium inside the cell. Okay, so concentration of potassium ions increasing inside the axoplasm, and the concentration of sodium ions is increasing outside. But we're also getting a charge. Okay, so this is creating what's called an electrochemical gradient. So electrochemical gradients are gradients like a concentration gradient, but also to do with charge. All right, so we've now got it slightly more positive on the outside and slightly more negative on the inside, because there's fewer positive ions on the inside here than negative ions, and there's now more positive ions on the outside here than negative. Okay, but also we've got a concentration gradient of higher concentration of potassium inside the cell than outside. So some of the potassium ions diffuse out of the axon down their electrochemical gradient. Okay, so they're going down, it's higher concentration of potassium, and it's there's a higher concentration of potassium here, so they're going to diffuse out. Now, diffusion of potassium ions is actually going to be limited. It won't eventually go back to equilibrium, because remember, it's also positively charged out here now and negatively charged here. So actually this movement down the concentration gradient is actually being counteracted by the charge. They're sort of cancelling each other out a little bit. Okay, but some potassium ions will diffuse out down their electrochemical gradient into the tissue fluid. Okay, because some of the potassium gated channels are open. But there's also an electrochemical gradient for sodium ions, and actually both the concentration gradient and the charge gradient are from the tissue fluid into the axon. So the sodium would diffuse, if it could, into the axon down its electrochemical gradient, because it's both positively charged out here and negative in here, so the sodium ions will be attracted to opposite charge, and there's a higher concentration of sodium ions outside than inside. However, because the voltage gated channels for sodium are closed, sodium ions cannot re-enter. Okay, the sodium potassium pump keeps going. Okay, some potassium moves out, but not all the potassium that's pumped in, but only some of it. So over time we establish this resting potential with this high concentration of sodium outside and the high concentration of potassium inside, okay, and a negative charge on the inside compared to outside, and this is the resting potential. It is a voltage. All right, so those are your key ideas. Okay, so what I'd like you to do, write your own set of bullet points to explain the resting potential. The next slide has also got a little summary that might be useful. I want you to think about why the term resting potential could be misleading. Think about the organelles you'd find in large numbers in the axon and why. Think about what I've said in terms of this process and how active it is. And suggest what would happen if a metabolic poison like cyanide was used on these cells. Okay, so you can pause the video there, have a go at writing your bullet points, rewind, have another look, okay, or look on the next slide. So here's some points that might be useful as well. Okay, once you've done all that, have a look at this plenary question. So this is actually what happens during an action potential. So we've had resting potential, which was a voltage. Here's the graph showing the changes in the charge of the axon membrane. Here, look, we've got the resting potential of -70. What happens during one action potential here? Have a think about describing it. What could be causing this change in charge here? How could that axon be changing? Okay, think about the structure of the axon. Look back at the previous slides. What could be changing in B? What could be changing in C? Okay, and we'll talk about this next lesson, and we'll go through this in much more detail, but that's what I'd like you to think about. Maybe read ahead about ready for next lesson. Thank you.